Howdy, folks. Thanks for checking into Mr. Ulrich's landofbiology.com. I am Mr. Ulrich. In this notescast, we're going to be talking about the father of genetics, Gregor Mendel, and how he was able to figure out some of the basic understandings of the inheritance of characteristics by studying traits in peas. Sure, modern genetics began in the mid-19th century in an Austrian monastery, but we can look even further back. The Bible has stories about genetics. Jacob and Laban, and you can look up the specifics back in Genesis, but to paraphrase a much longer story, Jacob agreed to take care of Laban's goats for the price of some of the goats. He would keep some of the goats that had a particular trait. Unbeknownst to Laban, what he did is he separated the goats, and through some other kind of scientifically suspect means, made sure that most of the goats had the trait that would give them to Jacob, and so he ended up with most of the goats. Now, Gregor Mendel did something a little bit more scientific. He employed quantitative analysis through rigorous note-taking of cross pollination amongst peas and keeping very careful track and counting up the characteristics of the offspring in order to figure out the basic underpinnings of the laws of inheritance. Mendel's work with pea plants relied on careful manipulation of the process of pollination. So he would cut the male parts off of the some flowers before they would grow to term to keep them from generating pollen. He would bag the female parts of other flowers to keep them from being pollinated. And then he could control which pollen grains were fertilizing which female flowers by actually painting the pollen grains onto the pistils. And by doing this, he would generate a group of flowers of pea plants that would all have, say, white flowers. Whenever they cross-pollinated with each other, they always had white offspring, white flowering offspring. He would do the same with purple flowering offspring, and so that all of their offspring were purple flowering. And he called these the true breeding parents, or the pea generation, the parental generation. He would then carefully manipulated so that he would crossbreed those and raise the seeds and observe the traits in this next generation, which he called the F1 or first filial generation. He would allow those to self-pollinate, so he would bag each one of those. And so they would self-pollinate, and the next generation would be called the F2 generation. Flower color was only one of several different traits that Mendel looked at. He also looked at the color of the seeds, the shape of the seeds, the shape of the seed pod, where the flowers were located, and even the height of the plants. As he followed these traits from one generation to another, he noticed a pattern in his notes. When he took parents of two different traits, all of the F1 generation would have one of either parent's characteristics or phenotype. When they were allowed to self-pollinate, he always got pretty close to the same ratio of phenotypes between the two different P generation parents. It was always a 3-to-1. There was more of the ones that were present in the F1 generation, but there were a few. So that trait of, in this case, white floweredness skipped generations. So it was present in the parent generation, not present in the F1 generation, and then present again, albeit only 25%, but still present in that F2 generation. Through following the patterns of the inheritance of these traits, Mendel was able to figure out that what happens is each parent donates what he calls a factor. We now know them as genes, but each parent donates a factor to the expression of a particular characteristic, say flower color. And that factor can, in the case of peas, be either for a purple flower color or a white flower color. We call these different factors that code for the same characteristic alleles. Now, to put a little more modern spin on this, we know that these genes are organized onto chromosomes. At a particular locus on a chromosome is going to be a particular gene for a particular characteristic. And now we know that there are variations. There can be, in this case, either a purple allele or a white allele at that particular locus. This is the nature of homology. So this is what we mean by homologous pairs of chromosomes code for the same characteristics, though they may code for different traits because they might have different alleles on them. So they code for flower color or flower shape or pod shape or flower location or height in the case of peas, though they might code for different heights or different flower colors or different flower locations. Traits, therefore, are inherited as discrete units. You get one from one parent and one from the other parent, two alleles, and that's where the nature of diploidy comes from. Die for two, one from one parent and one from the other. It's kind of like having two editions of a particular textbook. Like you might have two biology textbooks. They're going to talk about the same stuff, but they're going to have different phrases, different words, different sentences that describe the same concepts. Back in Regents biology, we talked about the law of dominance, that when there are two different alleles present, one allele is going to mask the expression of the other allele. In the case of our purple and white flowers, that we don't end up with light purple when purple and white are crossed. We actually end up with all purple flowers, that the white allele is totally masked. We know now that that dominant allele actually codes for a protein that does something, that actually has a function. And the recessive allele is a gene that's broken. It might not even code for a protein at all. It might be so broken that it doesn't have the right sequence of nucleotides to even generate a protein. We often talk about the dominant alleles as the wild type, as most often alleles are there to code for something. And the mutants are the ones that have something wrong with the allele so that the protein doesn't work. And the resulting trait is the result of not having that protein. Well, now I've used the word phenotype a couple times, and we should probably define it here. The phenotype is how the organism looks. It's the expression of the genes that they have. This is the physical, the pH. That's what the phenotype comes from. The genotype talks about the actual genes that are there. So when we're talking about dominant versus recessive, that's where we're really talking about the genotype. When we start talking about crosses, we typically use capital letters and lowercase letters to show the dominant and the recessive alleles. We got to be careful, make sure that you're using dimorphic letters. P is probably not a real good one. When I print them out here in text form here, you can tell the difference between an uppercase and a lowercase. But when you're writing them on a piece of paper, it's good to choose something like a Q or something like that. That's really different between the upper and lower cases. So here in our purple flowering pea plants, we're going to use the uppercase P to show the dominant purple allele and the lowercase p to show the recessive white allele. So our true breeding purple flowering pea plants are going to have two dominant alleles, and the true breeding white flowering pea plants are only going to have recessive alleles. When we cross the two, get the big peas crossed by the little peas, all of the offspring, the only resulting possibility, since the dominant purple flowers are going to make gametes that have the dominant allele, and the recessive white flowers are going to make gametes that have the recessive white allele, all of the offspring are going to have one dominant allele and one recessive allele. To apply these terms to the classic Mendelian test cross, here's my true breeding purple flower plants and my true breeding white flower plants. Those are phenotypes because they're physical expressions. To talk about their genotypes, the purple ones are going to have two dominant alleles, and the white ones are going to have two recessive alleles, both homozygous. And all of the offspring are going to be heterozygous, one dominant, one recessive allele. When we let those self-pollinate, that's where things are going to start to get interesting. This is where Punnett squares come into play. Now, what we're doing with a Punnett square is we're actually making a matrix of possible gametic combinations. Since when the plants are going to go through meiosis, those homologous pairs of chromosomes are going to segregate, and each gamete is going to get one of each homologous pair. That means that each gamete is going to get one of each allele. So we're really looking at which alleles each gamete is going to get. So in the case over here of the male, it's either going to get the dominant allele or the recessive allele. And in the female eggs, they're all going to either get the dominant allele or the recessive allele. It's not very exciting, but there's a little variability here. And now it's just a matter of which alleles, which gametes are going to meet to fertilize. And since we assume there's an equal chance of either type of gamete being produced and either type of gamete doing the fertilization deed, we can just go through and do a 4-by here and see that we have a 25% chance of big P, or homozygous dominant. I have in this box a big P and a little p, but I also have another box with a big P and a little p. These are both heterozygotes. And then I have 1/4 of them are going to be homozygous recessive, 2 little recessive alleles. So here's where we get that phenotypic ratio of 3:1. These 3 are going to be phenotypically dominant. They're going to show purple flowers. Even those heterozygotes, they're going to mask that white allele. And the 1/4 of the offspring that have 2 recessive alleles, those are the only ones that are going to have the recessive white trait. So this gives us our 3:1 ratio in the F2 generation. So our genotypes, 25% homozygous dominant, 50% heterozygous gives us 75%. And phenotypically, we get that 3:1 ratio. Well, I've been using these terms, homozygous and heterozygous. We should get them into our notes. Homo is for the same. So when we're talking about homozygous, the zygote is the zygous part. So those homologous pairs of chromosomes have recombined. And at this locus, they're the same. So homozygous dominant, so both dominant alleles, or homozygous recessive for both recessive alleles. We also have the heterozygous individuals, hetero for different, and that's where there are different alleles for that location. The problem for us geneticists is that organisms can have 2 different genotypes for the same phenotype. So how are we going to figure this out? We'll do a test cross. A test cross is when we take that phenotypically dominant individual, we don't know what its genotype is, and we cross that with a phenotypically recessive individual since we know what their genotype is. The test cross is going to work by basically showing us which of the 2 possible outcomes took place. Which ratios did we end up getting? If we end up getting all of the offspring purple, then we know that it was homozygous dominant. If we get actually half of the offspring being phenotypically white, then we know that we're talking about a heterozygote. Let's take some time to talk about Mendel's laws in a little more detail. His first law has to do with segregation. Now, we talked about meiosis last couple videos. Take a look at those if you need to. But we know that homologous pairs of chromosomes segregate during meiosis. They're going to bring their alleles with them. So that means that each gamete is going to get 1 of each homologous pairs of chromosomes. Now, when does all of this segregation take place in meiosis? Of course, during metaphase 1, because that's when the tetrads actually line up. We also get the crossing over, but that's metaphase 1. Following one trait at a time is what we call a monohybrid cross. And Mendel did this with flower color and seed color and all those other several different characteristics that we talked about. Mendel also followed the inheritance of 2 characteristics at the same time. So looking at both seed color and seed shape, or pod color and flower color at the same time. For example, here's the dihybrid cross between yellow round peas. So we're looking at pea color and pea shape versus green and wrinkled peas. Again, same pea color and pea shape, just different pea color and pea shape. And you notice that in the F1 generation, all of the offspring were round and yellow. So meaning that now we know that that one parent generation, one of them was homozygous dominant for both pea shape and pea color, and the other one was homozygous recessive for both pea shape and pea color. In the F1 generation, all of them are going to be heterozygous for both traits. When we did the self-pollination, you get this kind of interesting and characteristic ratio of 9, 3, 3 to 1. What Mendel was trying to figure out with these dihybrid crosses is if genes, or in his case factors, since the term gene hadn't been really coined yet, if these factors, when they're inherited, come in units. Do they come together or do they actually segregate independently from one another? What this means is if you get a recessive allele from for one trait from that parent, do you get other recessive alleles or other traits from that parent as well as kind of like a unit, or do they independently segregate so that you have all different combinations of dominant and recessive alleles for either trait? So let's check it out, which way actually works. If they're hooked together, when you get a dominant allele in a gamete, you're going to get the other dominant allele in that gamete. When you get the recessive allele in a gamete, you're going to get the other recessive allele as well in this case. So the Punnett square is very simple. It's going to look just like we did for the monohybrid cross. And we'll get the same phenotypic ratio of 3 to 1. But what we actually see here from the results is a 9-3-3-1 ratio. So clearly this is not working. So let's look at the other way. Now, this is going to be a little bit more complicated because there are more choices. There are more ways that those alleles can arrange. I can either get the dominant Y and the dominant R allele, or I can get the dominant Y and the recessive R allele, or the vice versa, the recessive Y and the dominant R. I can get both recessive Y and R. So I have each one of those different types of gametes that are possible. Remember, a Punnett square is to work out the possible gametic combinations. When we start putting these together and fill out the Punnett square with all the possible combinations, we can see here are the 9-3-3-1 that we're trying to get to match, I should say, from the experimental results. The results of these dihybrid crosses led Mendel to develop his second law of heredity, one of his shakier ones, called the law of independent assortment, which states that different factors, which we now know are different genes or alleles that are located at loci, separate into gametes independently of one another. So if I have two traits like yellowness and greenness versus roundness and wrinkledness, and I have an individual who is heterozygous, they're going to go through meiosis, and in one way they can make different gametes. And if the chromosomes line up a different way, then they can make a different array of gametes. And there's an equal chance of all of those different gametes being produced. Now, this does work for chromosomes, or for traits, I should say, genes that are on different chromosomes for sure. So if they're non-homologous, then they are certainly going to align independently, and they are certainly going to be segregated independently from one another. And this is going to produce a wide variety. But there's actually even more variety than that. What we've actually noticed is that they don't even need to be on the same chromosome in order to be inherited independently from one another. There are so many chiasma and so much crossing over that takes place that, especially when we start getting further and further away from the centromere, crossing over is so routine and so regular that they're going to act as if they segregate independently from one another, even though they're located on the same chromosome. As with the law of segregation, independent assortment is also taking place during metaphase 1. The big exception here are those traits that are located really close to one another and close to the centromere, so that they can kind of get around some of that crossing over that shuffles the rest of the genes that are further away. These tend to be inherited together, and we call them linked. So traits like red hair and freckles, things like this that tend to be inherited together, are linked. The key to Mendel's success as a researcher, and the key to most researchers' success, was meticulous and painstaking note-taking and data crunching and data analysis, all the way from gamete formation all the way up to the breeding individual. Mendel then was able to determine the laws of segregation by doing monohybrid crosses, looking at those single traits and determining that each characteristic is controlled by an allele that segregates into different gametes. So each parent donates one of a pair of alleles for a particular trait. Independent assortment, which he determined through the dihybrid or even more crosses, showed that the segregation of alleles takes place independently from one another. We know now, of course, that there are these linked traits that are on the same chromosome, close together and closer to the centromere to avoid the shuffling of crossing over. Mendel's choice of pea plants as a test subject was a good one. There's lots of different traits that we can look at in pea plants that are obvious, not hard to see, flower color and pod shape. Plus, it's really easy to control which plants are mating with whom. We can bag plants, we can cut off anthers, we can paint pollen onto the female parts of other plants. Significant amounts of luck involved here. He also chose traits to follow that were basically a monogenetic that is controlled by a single pair of alleles. And he also chose traits that showed complete dominance and recessivity. As we're going to talk about in future videos, not all traits follow simple Mendelian genetics, but this was a fantastic start. And amazing that he was able to determine all of these things without knowing what DNA was, the importance of it, how it functioned, or any of the stuff that we now kind of take for granted. Oh, but we'll talk about non-Mendelian patterns of inheritance in the next video. For now, thanks for checking into Mr. Ulrich's landofbiology.com. Again, I am Mr. Ulrich, and if you have any questions, feel free to drop me an email, write them in the comment section below, any of those things. Other than that, we'll see you in class.